speaker is Gheorghe, and he will talk on the mechanism of bidirectional replication initiation in Metazoa.
So I'm really thankful to the organizers for giving me a speaking slot, especially because this is the first complete story coming out of our lab. So it's really an honor to be presenting here. So the previous speakers have given a really good introduction to initiation, so I can probably skip most of it. But I just want to emphasize, replication involves the loading of 2 inactive helicases at each origin, then the recruitment of the GINs and CDC45 subunits to form 2 helicases, then the activation of these helicases, and then subsequent bidirectional replication. So this process involves the participation of these proteins called firing factors who have mysterious roles and who don't seem to be involved in actual replication elongation. So the mechanism of initiation in Metazoa is really poorly understood for a variety of reasons. And our lab is interested in understanding how is bidirectional replication initiated with high fidelity at thousands of origins across the genome. And more specifically, we're interested in understanding how is this twofold symmetry enforced during initiation. So how are 2 GINS molecules recruited, how are 2 CDC45s recruited, and what is the function of these mysterious firing factors? What do they do, how do they do it, how many copies of these factors are required, and what order do they function in? So to do this, our lab has been optimizing and adapting a single molecule approach that I developed when I was a postdoc with Johannes Walter. And the idea is we use xenopus egg extracts, which are really good at replicating DNA. And what we do is we take DNA molecules, we stretch them, we tether them onto a flow cell, we license the DNA in an extract that mimics G1, and then we replicate the DNA in another extract which recapitulates S phase. And the key idea here is that we can deplete any protein of interest, for example, GINs, and then we can add it back, fluorescently labeled version of the protein, which floats around an extract, and then we can use single molecule microscopy to track the recruitment of this fluorescent protein, its incorporation into CMG, and then the bidirectional movement of helicases that contain this protein. And in addition, we can monitor DNA synthesis by adding a protein, FEN1, which binds to nascent lagging strands. And this enables us to look at about 1,000 molecules per experiment, 1,000 DNAs per experiment, and more importantly, we can visualize really fast and transient events that are really impossible to capture using traditional methods. Okay, so the project started in the summer of 2020, the glorious first summer of COVID. Scott was a really brave grad student who joined my lab, and we started looking at the recruitment of GINs. So what you see here is a chymogram from one of our experiments, and what we see is time goes from top to bottom. It's just like a projection of the movie. We see the recruitment of GINs, which is this green signal. This GIN stays for about 2 minutes, and then we see bidirectional origin firing. The blue signal is DNA synthesis, and we see that there are 2 helicases which contain GINs. The surprising thing was that the GINs was recruited in one step, so 2 molecules recruited at once within 10 seconds of the movie. And this was really puzzling because GINs is a monomer in vitro, we checked that. And we figured maybe GINs is recruited in a highly cooperative manner, and we just don't see this stepwise recruitment. So to resolve that, we began titrating GINs down all the way as far as we can go to about 5 nanomolar. And even at 5 nanomolar, we still see this simultaneous recruitment of 2 GINs molecules. So we see this for hundreds and hundreds of initiation events. And we ended up considering this model where somehow this GINs dimerization is enforced in solution by some sort of scaffold. And it's this scaffold with 2 GINs molecules that gets recruited, and that's what enforces bidirectional replication, or part of the mechanism that enforces bidirectional replication. But on top of that, we see some really, really interesting dynamics. So prior to the stable incorporation of GINs into helicases, we see these transient recruitment events that correspond to single GINs recruitment attempts, but they are immediately rejected. So it seems like the initiation machinery has a way of distinguishing 1 GINs versus 2 GINs, and it's rejecting 1. And another really interesting thing that we observed is, in addition to all these productive recruitment of 2 GINs, which lead to replication, we see lots of non-productive events where 2 GINs molecules are recruited, but they don't fire. And eventually they just photobleach, right? So they're very stable, lasting for many, many minutes. And this corresponds to about 50% of all 2 genes recruitment attempts, which is not insignificant. Of course, the next logical step was to look at CDC45, and Scott did that. And to make a long story short, it's pretty much the same thing. We see simultaneous recruitment of 2 copies of CDC45. We see these transient binding events of one CDC45 that get rejected. And this was really puzzling for 2 reasons. One is that CDC45 is a monomer in vitro, and we checked this. And the second one is data coming from the Bell lab showing that in the yeast reconstituted system, 2 copies of CDC45s are recruited sequentially to the origin. So I think this may underlie real differences between how yeast do initiation and how metazoans do initiation. And again, we're forced to consider this model where there's a dimerization scaffold which brings 2 copies of genes to the origin. And again, we see these non-productive 2 CDC45 recruitment events, which are quite common. And we think that these events may represent assembled helicases that fail to fire for whatever reason. Could we see them with genes, or we see them with CDC45? Okay, so we have these models. We wanted to figure out what are the scaffolds that facilitate this dimerization. And we were inspired by this paper from yeast, which showed that several proteins co-IP together from extract, but only in the presence of cross-linking agents. So the idea is that these proteins facilitate the recruitment of polyepsilon and genes to the origin, and our top 2 candidates were TOBP1 and RECUL4. So the idea is, whatever the scaffold is, it should arrive to the origin simultaneously with its cargo, right? So with genes or CDC45. And Ricky bravely, a postdoc in my lab, he bravely volunteered to do this. Okay, so we started with TOBP1. This is full-length TOBP1. We fluorescently labeled it. And what we saw is the transient binding and dissociation of TOBP1 a couple of minutes before the firing of the origin. Right, so this is DNA synthesis signal. So we call these productive events. But we also see a ton of non-productive events, binding dissociation, but no DNA replication. Now, the problem with full-length TOBP1 is this. So TOBP1 likes to form, call them condensates, aggregates, whatever, big assemblies that make it very difficult to acquire data and interpret it. So we switched to this truncated form of TOBP1, which lacks the ATR activation domain. And the Dunphy lab has shown that this form of TOBP1 is fully capable of initiating replication. So now the data is a lot easier to interpret. There's a lot less background. And we see, again, very clear recruitment dissociation events prior to origin firing. And in addition to these, we see a ton of non-productive events. And in fact, only about 5% of all TOBP1 binding events result in initiation. 95% are non-productive. The other interesting thing was that when we look at the productive events, a significant portion, about 40%, contain a single copy of TOBP1. And that was that was disappointing because we're looking for a dimerization scaffold, which should be a dimer. But it was also puzzling because there's lots of reports that TOBP1 should be able to oligomerize, at least in the context of DNA damage signaling. So, we looked at TOBP1 in vitro using in vitro photobleaching, and we saw that full-length TOBP1 as well as truncated TOBP1 are overwhelmingly monomeric. And consistent with this, even when we do see 2 copies of TOBP1, the 2 copies are recruited sequentially, not as a constitutive dimer, but one at a time. And finally, okay, so by this point, TOBP1 is not looking like a very good scaffold candidate, but we wanted to double check. So what we did is we measured the time delay between the binding of TOBP1 and the firing of the origin, and we find that on average, it takes about 3 minutes. And when we do the same thing for GINs, we find that the waiting time for GINs is about 2 minutes. So TOBP1 arrives before GINs, therefore it can't be the scaffold for GINs. And we moved on to our next candidate, RECUL4. Ricky did a similar series of experiments. The data is very similar, so you see transient binding of RECUL4 prior to origin firing. But unlike TOBP1, RECUL4 is very efficient. So most of the RECUL4 recruitment events are productive, more than half. And RECUL4 forms stable dimers in vitro, and we're very happy because this is very promising for a dimerization scaffold. And we did the killer experiment where we simultaneously visualized the recruitment of GINs and RECUL4, and we would expect them to arrive together. And to our great disappointment, we see that GINS, which is this green signal, GINS always or almost always arrives first, and RECUL4 arrives later. And we see this for dozens of molecules. So we concluded that RECUL4 is not the dimerization scaffold. Okay, so at this point, it's late 2022. We've rolled out 2 scaffolds. I was talking to Johannes Walter about our data and sort of trying to make sense of it, and he said, well, we have this protein, which is involved in initiation. AlphaFold predicts its volumes dimers. It interacts with GINS, TOPP1, MCM3, and I said, okay, this looks exactly like the thing that we're looking for, the dimerization scaffold. So Ricky bravely made all the reagents, and we see the same picture, this hallmark of firing factors, transient recruitment of Donson, dissociation, and immediately origin firing within a few minutes. And again, Donson is very efficient, so 2/3 of the events are productive. And the events that are productive are actually quite short-lived, only about a minute. And then they dissociate. However, the events that are non-productive tend to be much longer, and eventually they disappear because they photobleach. But when they photobleach, we can tell that there's 2 copies of Donson there. And we also check that it's a dimer using other approaches. Now, again, the killer experiment is simultaneous imaging of Donson and GINS, and they should be arriving together. And to our great satisfaction, they did indeed arrive together. So GINS, the green signal, is recruited at the same time as Donson. Donson leaves, GINS stays behind. You get replication initiation. And our data says that Donson does not travel with the replosome, right? There's no, all of these productive events dissociate. And in fact, our data says that when we see non-productive events, which are long-lived, as you can see here, GINS is recruited, Donson is recruited. For some reason, Donson stays bound for a really long time and eventually photobleaches. But those events never fire, which means that the release of Donson from the origin is a prerequisite for replication initiation. So failure to release Donson leads to no origin firing. Okay, so just to summarize, our data really gives this reveals a very dynamic picture of initiation. There are lots of failed attempts to initiate replication. We've identified several metastable states, and we think that these unstable states provide a mechanism for quality control. So you can retain 2 copies of GINS when they're recruited, or 2 copies of CDC45, and you can quickly reject when single copies are recruited. And we also have data, which I can't show you right now, is that CDC45 is recruited about 15 seconds before GINS. But we're not sure if this order is strictly enforced. And this 2-fold symmetry of bidirectional replication is supported by dimerization scaffolds, and we've confirmed that Donson is the dimerization scaffold for GINS, in agreement with everybody else who's talked earlier today. We don't yet know the dimerization scaffold for CDC45. We have a couple of candidates, but it's an ongoing project. We have very interesting data and insights into RECUL4 function, and we have data to show that it facilitates CMG activation, not CMG assembly. And the role of TOBP1, it seems like TOBP1 binds to the dormant origin early, and it acts as a lasso to bring in this complex of Donson and GINS. And that being said, I want to thank everybody in my lab, especially the people who did the work, and they all have posters, so you can go to them. They're all 3 separate sections. I'm really also thankful to Linda, who raised a lot of the early antibodies. I want to thank the Walter, Dunphy, and Booz labs for sharing insights and reagents, and Carlene and her lab for feedback and support as we were building up the lab.
Thank you very much. Any questions?
Hi, Gheorghe. That was great. Just so comment and a question about the non-productive events. I mean, I guess making CMGs that are not activated could be one reason why, I guess in the biochemistry we've heard about earlier, it can look as though Donson's associating with replisomes. They might be just associating with non-productive CMGs that haven't actually kicked off. And so a question there, just thinking about, what's going on with these non-productive ones, why don't they fire? Do you see any correlation between, because you showed a recruitment of RECQL4, is there any correlation between, which is a later event to GINS, correlation between that and whether you then have a productive event?
Right. So I cannot answer that question definitively, but I can show you some interesting data. So, okay, when we deplete RECQL4, we still, even though we deplete 99% of RECUL4, we can't see it on a Western blot, there's still residual DNA replication. And so basically, when we see, so if we deplete RECUL4 and we look at genes recruitment, we see that 90% of genes recruitment is like this, is non-productive. 2 genes are recruited and then they never fire. But a small fraction, about 8%, manage to fire despite the fact that we've depleted RECUL4. So either the tiny, tiny amount of RECUL4 left manages to participate, or they can fire without RECUL4. And what we see here is we see a long delay between the binding of genes and the origin firing. So this is a mock control, it's about 2 minutes, and the RECUL4 depleted experiment is about 5 minutes. So the absence of RECUL4 definitely prolongs the waiting time between genes recruitment and firing, consistent with the fact that RECUL4 does something downstream, presumably activation. But I cannot put my hand on fire and say RECUL4, RECUL4 is participating here. Maybe they can fire despite the absence of RECUL4.
But in the cases where you do see recruitment of RECUL4, as you showed us earlier, do they always go on to be productive?
In the case of RECUL4, I think it's more than half the events are productive. So where's the RECUL4? Most of the RECUL4 events are productive. Yeah. So and what you can see is RECUL4 sometimes makes repeated attempts to support origin firing at the same locus. Right? So it's trying to help this origin that will never fire. So there's something pathological about these guys.
Hi, Gheorghe.
Yes, hi.
I think you can do that experiment, but I'm not sure whether you did that or not. So when you deplete RECUL4, do you see Danson stay around the origin or not?
And we have not done that exact experiment now. It's something that we should try to do, yes.
Thank you.
Hi. I hope this is a simple question, but why is it so important to reject single loading events?
Well, I'm not the one who makes up the rules, right? I think, where is the model? I think it's important, well, it's an advantage to only accept dimers because dimers are guaranteed to give you a bidirectional origin firing. Why nature does it this way, I don't know, but it makes sense that you would reject single recruitment attempts. One interpretation is that maybe genes without any scaffold, just genes by itself can dock, but the binding is very weak and immediately gets rejected. So maybe it's genes binding without Danson. Or maybe it's a dimer of Danson with a single copy of genes, and again it binds, but it's on the wrong conformation, it just doesn't fit, right, and immediately gets rejected.
In the interest of time, thank you very much.